Okay, next we have the observation of the cell membrane in a rheole. We are having a lab activity here, cell membrane in the rheole. Okay, here, for this activity we have to take a rheole leaf and break it to get the peel of the leaf and take a permanent slide and put the peel, the rheole leaf peel on a slide and put a drop of water and cover with a cover slip and observe under the microscope. Then you will get the compartmental structures. Okay, so this type of compartment-like structure are the rectangular compartments are observed in the rheole leaf. So the cells look like a rectangular in shape. So with this, when you observe the cells of the cheek cells, okay, when you observe the cells in a rheole leaf, the cells look like a rectangular in shape, whereas in cheek cells we observe the circular. Okay, so while observing, by doing this lab activity, we can state that all the cells are not in a similar shape. Some are rectangular, some are circular, some are irregular in shape. Okay, and coming to the another one, cell size. The cell size also varies from organ to organ and organism to organism, so all the cells are not equal in size. Some cells are small. The smallest cell will be the mycoplasma, and the largest cell will be the ostrich cell. Ostrich egg cell. The smallest cell will be the mycoplasma cell, and the largest cell will be the ostrich egg cell, and the longest cell will be the nerve cell. Okay, this is the distinction of the cell. Some are small and some are large, and some cells are long. Okay, so here coming to the human being, the smallest human cell will be the sperm cell, and the largest human cell will be the egg cell or the embryonic cell. So this is about the distinction of the cell based on their size. So all the cells are not equal in shape and not equal in size. Okay, this is about some of the information about the cell, and next we discuss about the cell organelles. So what are they? The cell organelle. For this, we are also having one activity. Okay, another activity is the observation of nucleus in the cheek cell. As earlier we stated that the nucleus is the control point of the cell, so we will observe the nucleus by an experiment. So for this we require the toothpick and a permanent slide and cover slip and microscope. And for this, we will take the help of one stain, that is one dye, that is the methylene blue. So you have to remember what is the dye used in staining the nucleus. Means here, this is the methylene blue. Okay, for this activity, we have to take a fresh toothpick and put it into the mouth and scrape the inner lining of the cheek 4 or 5 times. Then you will get some matter or some material to the toothpick. Then put it on a permanent slide. Then pour one or two drops of the methylene blue on the collected cheek lining material and put it, spread it equally on the slide. Then excess of stain will be removed after keeping it for 4 to 5 minutes. Then put a cover slip and observe it under the microscope. So when you observe it under the microscope, you will get the image like this. Okay, when you observe it under the microscope, you will get a dark boundary that is called the plasma membrane. The dark boundary will be the plasma membrane. Dark blue boundary is the plasma membrane. And the blue colored fluid matrix, that is called the cytoplasm. And next, dark blue colored circular structure present in the center, which is a dark blue in color, that will be named as the nucleus. So with this activity we can able to identify the nucleus of the cell. We are taking the help of the methylene blue. Okay, so we have to take few precautions for doing this activity. First precaution will be that don't scratch the cheek so deeply, it may injure the blood vessel. Then second one will be that spread the cheek cells uniformly on the slide and next try to avoid the formation of the bubble on the slide. So these are the few precautions we have to take for doing this activity. With this we can able to observe the nucleus in the cheek cell. Okay, this is about a few information regarding the cell membrane observation and cell membrane in the rheolith and nucleus in the cheek cell. Okay, we will discuss a few points about the nucleus. So, nucleus is a control point of the cell. It is present exactly at the center in the animal cell, whereas in the plant cell, it is somewhat sideways from the center. And this is the largest organelle of the cell. And coming to the another one, why it is called as the control point of the cell. Means here, all the genetic material which is required, all the genetic material which is required for the formation of the various organs and organ system is coded from the nucleus only. So all the transcription of the genetic material and the genetic coding, all this process takes place from the nucleus. So the genetic material is there in the nucleus and in the chromosomes. So whatever the character of the organism, so must come or must transcribe it from the genetic material of the nucleus. And another one here, there is a boundary which separates the nucleus from the cytoplasm. So that is called as the nuclear membrane, and the fluid matrix inside the nucleus is called as the nucleoplasm. So there is a clear-cut separation of the nucleus and the cytoplasm in the eukaryotes, whereas in prokaryotes all the nuclear material is scattered in the cytoplasm. So there is no nuclear membrane in prokaryotes. So this is about the nucleus, which is a control point which regulates the various biochemical activities of the cell, and all the genetic information is stored in the nucleus of the cell. Okay, next one will be the endoplasmic reticulum. Endoplasmic reticulum is a multi-layered membrane which starts from the plasma membrane throughout the nucleus. So it is a transport channel between the plasma membrane and the nucleus. So it is a multi-layered transport channel arises from the plasma membrane. So when you observe the endoplasmic reticulum, it will look like this here. Okay, so here a network of membranes. A network of membrane arises from the plasma membrane, and this is the transport channel for the passage of the various material from the nucleus toward the plasma membrane, or from the plasma membrane toward the nucleus. And here we will observe the oval-shaped granules or the circular granules on the surface of the endoplasmic reticulum, which are called as the ribosomes. Okay, so here the circular granules present on the surface of the endoplasmic reticulum are called as the ribosome. So here, based on the presence or absence of the ribosomes, the endoplasmic reticulum exists inside the cell in two forms. One is a rough endoplasmic reticulum and a smooth endoplasmic reticulum. Okay, rough endoplasmic reticulum possesses the ribosomes, and these ribosomes are the main organelles for the synthesis of protein. So the main role of rough endoplasmic reticulum is the protein synthesis. And next one will be the smooth endoplasmic reticulum, which is devoid of the ribosomes. Okay, here the smooth endoplasmic reticulum doesn't possess any granular material on its surface. That's why it is called the smooth endoplasmic reticulum. And what is the role of the smooth endoplasmic reticulum means here? It helps in the synthesis of lipids and fatty acids. So lipids and fats are synthesized in the smooth endoplasmic reticulum, whereas the rough endoplasmic reticulum takes an important role in the synthesis of protein. And one more important function of the smooth endoplasmic reticulum in the liver cell, it plays an important role in the detoxification reaction. It plays a key role in the detoxification reaction, the smooth endoplasmic reticulum of the liver. Okay, so this is about the endoplasmic reticulum. It is a multi-layered network of the plasma membrane, which is the transport channel, and it helps in the synthesis of proteins, lipids and fats. And it also takes an important role in the detoxification reaction. Okay, next one. This endoplasmic reticulum is having another structure. We will discuss about another structure. This endoplasmic reticulum, the number of endoplasmic reticulum may vary from cell to cell, which the number will be not limited in a cell. So these are scattered in the cytoplasm. Okay, next one will be the Golgi complex. The Golgi complex or the Golgi bodies. This is a network of vascular structures, vesicles or vascular membrane structures which are there in the cytoplasm like this here. Okay, Golgi complex or the Golgi bodies possess a membranous, multi-layered membranous vesicular structures and which also possess vacuoles, lysosomes and all they are in the Golgi complex. The main function of this Golgi complex is the packing and transportation of the materials which are synthesized in the endoplasmic reticulum. So here, whatever the proteins, lipids are synthesized in the endoplasmic reticulum are packed and transported to the different cell organelles only through the Golgi complex. And this Golgi complex is having two parts here. One is the convex end is the forming end and the concave end is the secreting end. Convex end is the formation end and the convex end is the secreting. Convex end is the formation end and the concave end is the secreting end. Secretory end. Okay, here as the material synthesized in the endoplasmic reticulum are collected by the convex formation end, whereas it is secreted or transported from the concave end. This is called as the secretory end. So, meanwhile, the Golgi complex is a main organelle of the cell which has the packing and transport of the various material across the cell. Okay, next we discuss about another one here. Plastids. Okay, plastids are the organelles, oval-shaped organelles present only in the plant cell. Okay, these plastids are again of three types here: chloroplast, chromoplast, and leucoplast. Okay, plastids are the oval-shaped structures scattered in the cytoplasm, and these plastids are again of three types here: chloroplasts, which are green in color, and these chloroplasts are absorbed in all the green parts of the plant, like leaf and stem parts. So, green parts of the plant possess the chloroplast, and this chloroplast possesses a pigment called as the chlorophyll. This is a light trapping pigment. This chloroplast plays an important role in the process of photosynthesis. When you observe the structure of the chloroplast, it is an oval shape, a bilayered organelle, and we observe a disc-like structure inside the chloroplast which are called as the grana, and the fluid-filled matrix is the stroma. Fluid matrix of the chloroplast is the stroma, and the disc-like structures are called as the grana. So here, the chloroplast is the main organelle in the plants for the process of photosynthesis. And next one will be the chromoplast. Chromoplasts are the colored plastids, which are red, yellow, orange, like various colors are there in the chromoplast. And these chromoplasts are absorbed in the fruits, petals and flowers of the plant. And next one, leucoplasts are the plastids which are absorbed in the seeds and grains of the plant. Seeds and grains of the plant possess the leucoplasts, which are non-green in color, which are the colorless plastids, like these are the colorless plastids absorbed in the various seeds and grains, and the chromoplasts are absorbed in the flowers, petals and fruits of the plant. And this chromoplast, oil soluble, the pigment chromogen is there in the chromoplast, and this is about the plastids absorbed only in the plant. And next we discuss about another topic, ribosomes. Okay, ribosomes are the circular granules present on the surface of the endoplasmic reticulum, and these ribosomes are present absorbed in the both prokaryotes as well as the eukaryote. The main function of ribosomes is to synthesize the proteins which are required by the organism. Proteins are the building blocks of the organism, and these proteins are synthesized from the ribosome. And another one will be the lysosomes. Okay, lysosomes are the suicidal bags of the cell. Lysosomes are also scattered. These are the vascular structures observed in the cytoplasm of the every cell, where these lysosomes keeps the cell clean by digesting the worn-out, cellular material. Worn-out cellular material, and it causes the digestive enzyme. Okay, thereby it keeps the cell very clean by destroying the entered pathogens. The pathogens which are entered inside the cell are catabolized or degraded by the enzymes released by the lysosome. Or if the cell is damaged, the lysosome itself, it releases the enzyme and destroy the cell. So that's why the lysosomes are called as the suicidal bags of the cell. Okay, and another one will be the vacuole. Okay, vacuoles are the storehouse of the cell, where it stores all the required materials of the cell. These are the large vesicular structures, and it stores all the required materials by the cell, and it also stores the waste material produced in the various metabolic activities and transported to the sites of the excretion. So vacuoles are the storehouses of the cell. And next one we will discuss about the mitochondria. Okay, here mitochondria is called the powerhouse of the cell. These are the tubular or the oval-shaped structure, which are green in color. For observing the mitochondria also, we are having a lab activity, observation of mitochondria in the onion peel. For the observation of mitochondria, we are using a strain Janus green bee. For the observation of mitochondria, we are taking the help of a strain Janus green bee. For this, we have to take the onion peel and put it on a permanent slide and pour 1 or 2 drops of the Janus green bee and the onion peel. Keep it aside for 5 minutes and then put a cover slip and observe it under the microscope. So when you observe it under the microscope, you will get the structure like this here. Okay, when you observe it under the microscope, you will get the central circular portion that is called as the nucleus, and the ovoid or cylindrical structure that is called as the mitochondria, which is green in color. For staining the mitochondria, we are taking the help of a Janus green bee. Okay, why the mitochondria is called as the powerhouse of the cell? Means here, this is the main site for the cellular respiration. All the cellular biochemical reactions are the energy produced in oxidation of the glucose, amino acids and fatty acids takes place inside the mitochondria of the cell, and the energy molecule and energy metabolites are produced as a result of cellular respiration in the mitochondria. That's why the mitochondria is called the powerhouse of the cell. When you observe the internal structure of the mitochondria, it is a cylindrical ovoid shape. A bilayered membrane is there for the mitochondria. The outer membrane and the inner membrane folds to form crystal. Outer membrane is smooth and the inner membrane folds to form a crystal, and it also in between the outer membrane and the inner membrane a fluid matrix, that is the cytosol. And this is the site for the various cellular reactions, and as a result of various reactions taking place in the mitochondria, energy will be produced and stored in the form of the ATP. That's why the mitochondria is called as the powerhouse of the cell. Okay, this is about the mitochondria, lysosomes, vacuoles and ribosomes. Okay, next we put a glance on the history of the cell theory. Cell theory. Cell theory was proposed by the Schleiden, botanist, and Schwann, a zoologist. According to him, all the living organisms possess the cell, starting from the unicellular to the multicellular organisms. All the living organisms must and should possess the cell. This will be explained by the cell theory proposed by the Schleiden and Schwann. This cell theory was proposed 200 years later, the discovery of the cell by the Robert Koch. And it also doesn't give clear-cut information about how the cells are formed. Then came into existence like Schleiden. And later Rudolf von Virchow. Rudolf von Virchow was the scientist who given a finishing touch to the cell theory and proposed the two postulates. According to him, here all the organisms possess the nucleus and its products. All the organisms possess nucleus and its products. All the cells possess the nucleus and its products, and new cells are formed from pre-existing cells. Okay, these are the two postulates of the cell theory proposed by the Virchow, and according to him, here all the cells possess the nucleus and its components, and new cells are formed from the pre-existing cell. And another one here, according to our assumption, the cells look like a flat, but the cells are not flat. They also have a length, breadth and width. So they also have the length, breadth and thickness. So that will be possible to observe the thickness of the cell in a low contrast microscopy. In a low contrast microscopy, we can able to get the information or the idea about the thickness of the cell. Okay, this is all information about the cell, its organelle, starting from the cell membrane, cell wall and various organs. So here, typical cell. So these are all the components present in the typical plant cell or typical animal cell. So what is a typical cell? Typical cell is a model which exhibits all the organelles present in the cell. Okay? So when you take the typical plant cell may exhibit all the cell organelles present in the plant cell. And typical animal cell possesses all the cell organelles present in the animal cell. For example, all the plant cell doesn't possess the chloroplasts. Okay? Why? Because chloroplasts are present only in the green parts of the plant. So, but typical plant cell represents all the components of the cell. Okay, so this is a model to study the internal organization of the either plant cell or animal cell. Okay, thank you one and all. Once you review all the topics of this cell, its structure and function, and we will come with another topic in the next session. Thank you.